Hmm?
Hello dear student, today I will be discussing into a new topic of receptor. Last day or last class I have discussed about the classification based on transduction mechanism by which receptor generally show their action. In that I divided or classified receptor into 5 types, that was G protein coupled receptor, then enzyme linked receptors, then Jack state receptor, receptor controlling transcription factors or gene expression, and ion channel receptor or ligand channel receptor. So in last class I have already discussed about GPCR, that means G protein coupled receptor, and you have to remember in our body most of the receptor, almost 60 to 70% or 80% you can say, they are comes under GPCR or G protein coupled receptor, and other 20 to 30% receptor they comes under remaining classes of receptor, that is enzyme link receptor, Jack state, transcription factors expressing receptors, ion channel receptor, etc. Like that, like last class what we have discussed about GPCR, like same we will discuss today about enzyme ring receptor, that is the second part or second classification of receptor after GPCR. So that is also called as ELR or enzyme linked receptor. So we have to now discuss about what is ELR or enzyme receptor or enzyme linked receptor. So we already know what is enzyme. Enzyme is catalyst. What catalysts do? Catalysts generally fasten or just speed up any reaction, but they don't involve any reaction, or they doesn't change their structure or their chemical form or anything in this reaction. What they did, they only catalyze or fasten up or speed up the reaction. Here also same thing, and receptor we have discussed that are chemical factors that generally helps drug or any other internals, like neurohormones, then growth hormones, other hormones and neurotransmitter to show their action into the body. So first we have to check what is the structure of ELR. Here I have simply and simplifiedly draw a diagram of enzyme ring receptor. So just see here, that is membrane. That membrane is called as transmembrane, and in that transmembrane, that is called as the receptor, and they present as receptor. As they are present inside or in that transmembrane, that's why they are also called transmembrane enzyme linked receptor. So here I have diagram or draw a simple diagram. Here that is transmembrane. In transmembrane, that receptor are present. So you have to check, you have to see, or you can see by the diagram, in that membrane inside part is intramembranal part, and external part is called as external domain or external membranal domain. So you can see that receptor, some part is present into the intra or the inside the membrane, and some part is present outside the membrane. Outside membrane is called as ligand binding site. So here generally ligand comes and bind with that receptor, and intra part, in intra whichever whatever receptor part is there, that part generally linked with enzyme, so that part is called as enzyme linked domain. So outside generally ligand will come and ligand generally bind with that part of receptor, so that is called as ligand binding domain. And inside intra part where cytosol is present, there generally enzyme will come and will bind with receptor, so that entire domain is called as enzyme link domain. So now we have to discuss about how, or the mechanism of action, how that receptor, enzyme link receptor show their action. So first of all, we have to study how they works. You have to remember, when enzyme link receptor they work together. That means if one receptor is present here, then another receptor will be present together, because they are together only will help each other and will show their action, and that will help overall enzyme link receptor to show its action. So before that you have to remember, in that from name only you can understand that with that receptor different enzymes will come and different enzymes will bind. I told you already, enzyme will bind into the intra part. Here which type of enzyme generally occurs, or which type of generally enzymes come and bind with, that is called as tyrosine enzyme. Most of the tyrosine enzymes, especially tyrosine, comes and bind with the enzyme linked intra-domain and bind with that enzyme ring receptor, and after binding they form a protein kinase that is called as receptor tyrosine kinase, that is also called as RTK. What that RTK will do, that RTK I already told you that in enzyme linked bind domain, that tyrosine enzyme will come and bind with a receptor and will form a complex that is called as receptor tyrosine kinase or RTK. That RTK what will do, that RTK will bind with that receptor already present into the transmembrane. So both receptor are present together. So what now happen, one RTK will come and sit into that receptor and another one will come and sit into that receptor, and by that binding with each other they will show their action. And how they will show their action, I will show you into the next diagram. Just suppose that is one enzyme link receptor and another more enzyme link receptor is present here. I have already told you that RTK, or that is called as receptor tyrosine kinase, that will come and bind with that receptor. That will come with receptor. When they will come receptor, what will happen in that intra or enzyme link domain, then that tyrosine protein or tyrosine enzyme will come and bind with that portion of the receptor, like this. I told you when that receptor will work, they will work together. Means when they will show their action, they have to be come together and they have to show their action together, or they have to help each other so that overall enzyme link receptor can show their action. Like here I told you, when they are coming together and present each other near, the tyrosine will come and bind into that enzyme linked domain of that or internal domain of that enzyme ring receptor. Like that here also tyrosine is present. So tyrosine is present. When the tyrosine is present, what tyrosine will do, that will activate RTK. That RTK will activate the tyrosine. When the tyrosine is going to activate it, they will help in the phosphorylation of ATP. So whenever they are helping in phosphorylation, what will happen? ATP will convert into the ADP, or that is called as adenosine diphosphate. How it will come? By releasing a phosphate. So what happens when that receptor are coming together, that RTK will come and bind with a receptor. With together that time only tyrosine will come and bind with the enzyme linked domain or enzyme link binding domain or intradomain of that receptor, and will bind with the tyrosine. When tyrosine is binding with that enzyme linked domain, that will help into the phosphorylation of ATP or adenosine triphosphate. For that phosphorylation, ATP will converted into the ADP by releasing one phosphate, and that ATP will converted into that ADP. Whenever ATP is coming into the ADP, that cause the activation of different protein. So what that will do, for the phosphorylation, ATP is converting into ADP, that will cause the activation of different protein. So whenever protein is activated, what it will do, that activated protein will come and bind with that tyrosine. So what now will happen that tyrosine protein or tyrosine enzyme will bind with that protein. That can be different protein that we will discuss into the other classes. Different protein whatever present in our body, they will come, they will activate that tyrosine and they will bind with tyrosine and they will form a tyrosine protein complex, and for that formation of tyrosine protein complex, that will cause the activation of different intracellular pathways, and that intercellular pathway at it, what it will do, it will do activation of transduction pathway, and for that transduction pathway, what it will help, it will help into the expression of gene transcription. So by that, receptor will carry that signal, and by that signal that will send that into that other cells. Like that, that drug will go and reach into the specific site and show that action. So what is the conclusion here? Here in transmembrane, enzyme link receptors are there. They have to be come together and they will show their action together. Whenever they have two domain, one is enzyme link domain, another one is link external or ligand link domain. Generally, which enzyme will come and bind? That is tyrosine. That tyrosine will bind into that enzyme linked internal domain of the receptor, and that RTK, that is called as receptor tyrosine kinase, that format will come and form, that will bind into that part. When that RTK will bind with that receptor, it will cause the activation of tyrosine enzyme. That tyrosine enzyme activation will leads to the phosphorylation of ATP. That means that ATP will converted into the adenosine diphosphate by releasing or secreting one phosphate. That activation of ATP to the ADP will cause the activation of protein, and protein that will come and will bind with the tyrosine into the enzyme linked internal domain, and they will form tyrosine protein complex activation. That activation will leads to different pathway that is going on into our intercellular. So that protein and tyrosine binding will leads to the activation of intercellular different pathway that will help into the expression of gene expression or gene or transcription, and by that they will show their transduction action, and they can show their action into the specific site with drug. So now I have to discuss about example what is comes under enzyme link receptor. Generally, growth linked factors or growth factors receptor comes under enzyme linked receptor. Example are fibroblastic growth receptor and insulin receptor comes under enzyme link receptor. So by that I have easily explained you what is ELR, what is enzyme link receptor, what is its structure, how it is going to show their action, what is their easy or understandable diagram, how they are working, what is RTK, what is tyrosine activation, what is ATP activation, how phosphorylation is taking place, how intercellular pathways are excited or activated, how gene expression is showing, how transduction mechanism is shown by ELR. All that I have already explained in that video. So by that I can think that you all understand enzyme link receptor very easily, because I explain here very easily how I can explain you. That is the easiest method of explanation, and I hope you all understand what is ELR, how it is going to show its action, how it is going to bind with different drug, how it is going to show transduction mechanism, etc., and what are the example or what are the receptor comes under enzyme ring receptor. Thank you.